Hi Biology, this is Mrs. Kozin. This video will be titled, this is Unit 2, Biochemistry, and this is Video 1, Chemistry Review. So today, in your notes, you're just going to be writing down a quick review of chemistry concepts, things that you learned last year in Chem 1. We're going to start with objectives for the unit. You guys do not need to write these down, just so you know some of the goals, the things that we're hoping to do by the end of the unit. First, we want to be able to describe the composition of 4 major organic macromolecules. Also, we need to explain their structure and function. The last one on this page, we're going to describe how they come apart and are built up through processes called dehydration synthesis and hydrolysis. The last two, we're going to be able to recognize and explain how these macromolecules contain a lot of energy in their bonds. And last, explain the role of enzymes, which are a very, very special protein that we will learn about at the end of our unit. So you do not have to write down any of those objectives. Those are just for us to review when we're looking at whether or not we've met our goals, if we've learned the objectives that we have set forth. This also, you guys don't need to write down. This is key vocabulary that you will see in this section of the notes. Many of these you have seen before. Again, this is just a quick Chemistry 1 review. Starting with structure of an atom. Subatomic particles. Remember, sub means beneath or smaller than. So subatomic means smaller than an atom. It's made up of 3 particles: protons, neutrons, electrons. Protons are positively charged, neutrons are neutral or have no charge, electrons are negatively charged. Inside the nucleus, you will find protons and neutrons. Also, you will find most of the mass of your atom. Electrons are orbiting outside the nucleus in the electron cloud. Element. So now that we know the structure of an atom, let's talk about what happens when we put multiple similar atoms together. The definition of an element is all one type of atom. So if I have 100 billion gold atoms, I have a sample of gold, that means it's all one type of element. Okay, all the same element, all the same atom. Now an isotope is also part of an element, but they're just different versions of the same atom. Now this example, we can tell that it's hydrogen. Remember, hydrogen's number on the periodic table is 1 because it has 1 proton and 1 electron. 1 is hydrogen. We can also tell this is hydrogen. 1 proton, 1 electron. This third one, also hydrogen. 1 proton, 1 electron. You can see the difference is that these have different numbers of neutrons. Remember, neutrons have a neutral charge. All they add to the atom are mass. So isotopes really are just fatter versions of the same atom. If you remember the example I talked about in class, my name is Mrs. Kozin. Even if I gain mass, I still remain Mrs. Kozin. That's the definition of an isotope, an atom that has gained mass, but its charge has not changed. Now, when we put elements together, chemically, we get what's called a compound. 2 or more different elements. So water, H2O, 2 hydrogens, 1 oxygen, that's a compound. NaCl, a sodium and a chlorine, those are chemically combined, that's a compound. Okay? And then when you put compounds together, you get what we call molecules. A molecule is more than 1 atom put together. Now you can have a molecule that's also an element. For example, if we have O2, it would be 2 atoms of oxygen, but it would still be considered an element because it's pure, it's all oxygen. Whereas when we put oxygen in H2O, it's still a molecule, but it becomes a compound. All right, now bonds. We have ionic bonds and covalent bonds. Ionic bonds be thieving, my friends. They're thieves, they steal. The ionic bond is where 1 electron leaves an atom or is transferred to another that takes it in. A covalent bond is sometimes a stronger bond that shares electrons. Many of the bonds that we will talk about in macromolecules are these covalent bonds. If you see my little comic here, take a peek at that, decide if you think that's an ionic or a covalent bond in that one. If you thought ionic, you're correct. This guy in the red says, I'm hit, I've lost an electron. So he's lost an electron, it was transferred somewhere else. And his buddy here asks him, are you sure? And he says, I'm positive. Because when you lose a negative, you become positive. Little science humor for your day there, my friends. All right, now chemical reactions. Just an overview. Remember, the materials that go into the reaction are called reactants. Think of it like the ingredients for a recipe. Chemical reaction happens here over the arrow, and then the products are what you come out with at the end. If you think of it like a sentence, here's the beginning, here's the end, the products are near the period of the sentence. Okay, now we're going to talk about how water bonds. Again, you don't need to write down this key vocabulary, just take a peek at it and think about maybe where you've seen it before we move on. We're going to talk about polar bonds, hydrogen bonds, acids, bases, and buffers. All right, acids and bases. Just a very quick review of acids and bases. pH scale, you guys, goes from 0 to 14. Right smack dab in the middle is neutral. Living things like to be neutral if they can help it. Below 7 is an acid. The closer you are to 1, the stronger the acid is. Closer you are to 7, the more neutral you are. Living things like to be near 7. Base is higher than 7, goes all the way up to 14. Now, think of some other examples of materials that living things would want to be near a 7, like pond water or tap water that we drink, or milk, or blood, or urine. Those are all things that living things want to be close to a 7. Remember, acids and bases that are very low or very high on the scale, they can be very corrosive and they can burn. So obviously living things don't want to have blood or milk or urine that burns. We want to be closer to 7. Now, a buffer is something that we can, our body can create to help us prevent sudden changes in pH. All right, so don't forget, you guys, right there, that living things like to be near neutral if they can help it. Polar bonds, okay? These are not bonds in the Great White North, my friends. These are bonds that are created by opposite poles. As a molecule becomes charged, what that means is all the protons like to move to one side, which makes one side positive, one side negative. And just like a magnet, your opposite sides are attracted to each other. Opposites attract here in polar bonds, my friends. So you can see right here in my little comic, we've got the teacher asking perhaps one of you gentlemen would mind telling me just what you outside the window is so attractive. You can see the polar molecules being attracted to the positive molecules being attracted to the negative. So that's a polar bond. And last one, example of a polar bond that's used in water is a hydrogen bond. Here's a lovely water molecule, okay? H2 and an O. Now my electrons are all migrating to one side, and so that makes this side negative and this side positive. And just like lovely little Legos, our water molecules are going to pile right on top of each other with the positives and the negatives attracted to each other. This is the thing that helps to hold our water together. And given you guys that we're made of between 75 and 80% water, it's pretty important that our water holds itself together. Okay, so that is hydrogen bonds. And that is the end of our first notes. Unit 2, Biochemistry and Chemistry Review.